We are going to be talking about genomes, genes, and the genetic code. And this topic has, it's kind of a short topic with lots of definitions and things like that that will spill over into the next coming few topics. So remember that I had mentioned that for the final exam, there are 3 things that you should know lots about. So we have replication, which we talked about this week. Next week, we're going to talk about transcription and translation. So we're going to talk a little bit about transcription and translation today, but we want to talk about genes and genomes and things like that first. First thing is, what is a genome? We've kind of thrown the word around a few times already. I'd mentioned, for example, that mitochondria have their own genomes and chloroplasts have their own genomes. So genome is all the DNA in a cell or an organelle. It doesn't just include genes, it includes other things. So some structural components that we've talked about, for example, we've talked about origins of replication. We've talked about telomeres and we've talked about centromeres. These are not genes, but they are part of the genome, part of the DNA that's in a cell. Also, something we're going to talk about here eventually are things like regulatory components, and we'll talk about that. I think that's topic 18. So there's quite a bit to the DNA other than just the genes in general. So we've also discussed, what's going on in different types of cells. We talked about prokaryotic genomes. They have a circular chromosome, and so, for example, E. coli. That represents somewhere around maybe 4,300, maybe 5,000 genes, somewhere in that category. There are some organisms with only about 1,000, and there are some microorganisms with much larger genomes. Usually we're looking at a single copy, and we don't usually necessarily describe these organisms as diploid or haploid, but technically they're haploid because they only have one copy of their genome. And they also have sometimes, not always, but sometimes prokaryotic organisms have plasmids. And these are kind of like mini chromosomes, and they'll have a few more genes. So pretty typical plasmid might have 3 to 5 genes. Some have many more. I think some of the biggest ones have about 100 genes, but those are pretty unusual. So eukaryotes, we are much more complex. We have multiple chromosomes. Usually there are a few weird protists with only like 1 chromosome, but sometimes we talk about ploidy, so we might be haploid or diploid. Some organisms, plants and reptiles, can be tetraploid, and there's all sorts of different things that can go on in terms of the genomes of these organisms. This is showing the genome of Drosophila. So Drosophila is the scientific name for the fruit fly. And it's a favorite of geneticists because these organisms are pretty easy to grow and they're cheap. And so we know a lot of genetics from studying fruit flies. So they have 4 pairs of chromosomes, so 8 chromosomes. And you can see that, most of them are relatively large. And then they've got these 2 little ones here. I don't know a lot about Drosophila genetics, but in some ways they're similar to humans, where they are diploid organisms, just like us, and they have haploid gametes. We talked about the human genome. We talked about the fact that we have 46 chromosomes or 23 pairs. So I think I showed you this picture before, and you can see from here that this person is XY. So this person is probably a male. This isn't necessarily the complete human genome because this doesn't include the mitochondrial genome, which would be all of the DNA in a cell. But this is the, I guess, the complete nuclear genome, I suppose we would call it. So I want to talk about this. This is, if you think about this, this is really, really incredible. I'm always amazed by how much information is stored in a genome. So if you take a look at this, this is about somewhere around 20,000 genes, okay? This is, I think it's about 3 billion nucleotides. I have a slide on that. Here's the information here. So this is 23 pairs of chromosomes, 6 billion base pairs. So that's an incredible amount of information. So you can think about our cells as being these information-rich things, much more complex than machinery or libraries or computers in some ways. So I just wanted to show you for comparison how much information is actually stored in one human cell. So the King James Bible, I looked that one up, and that is about 3.5 million letters. So not even close. There's way more information in your genome than is found just in one thick book, such as the Bible. If you're not as familiar with the Bible, maybe you're familiar with the Harry Potter series, which is 7 books, and that's about maybe 5 million letters. I was able to look up the words and how many words there were, but I just estimated how many letters. So not even close as well. So what about something bigger? What about a complete encyclopedia kit? Probably most of you aren't use these anymore. Every house used to have one back in the day, and they'd have one for each letter of the alphabet, have all these articles about different things, and Encyclopedia Britannica is somewhere around 300 million letters. So what do we mean for the human genome? It's a little bit more equivalent to the English Wikipedia. I haven't looked up how big Wikipedia is lately. It's possible it's even bigger now. But think about Wikipedia compared to a normal encyclopedia. Wikipedia has articles on Star Wars and celebrities and all sorts of other things that are not necessarily included in Encyclopedia Britannica. So huge amount of information in there. And I just think it's really incredible. I think that the human genome is a really cool thing. But this is not the end of the story. So here's the other question, and this is the thing that I find really even more mind-blowing, is what is in the code, right? What do those chromosomes actually code for? So it turns out that actually a small percent only code for genes. So we're estimating, current estimate is about 1.5%. So that means 98.5% of the human genome is coding for a whole bunch of other stuff. So what does this include? This includes something called introns. We haven't talked about introns yet. We're going to talk about introns in our next topic. So don't worry about those yet. Big percentage codes for regulatory sequences. So we're going to talk about gene regulation, but it's really important as complex organisms that we turn on or turn off the right genes at the right time. You don't want to be producing hemoglobin in your brain, right? We want the hemoglobin in our blood and so on. So there's a lot of this has to do with the regulatory processes. There's still a big chunk that's kind of unknown. Structural, of course, this would include things like centromeres, right? So centromeres and telomeres. But there's this big chunk that's kind of unknown. We used to call this junk DNA, but scientists got away from that because as we learn more of the genome, we learn that most of it's not junk. Most of it is actually good stuff. And we're hoping we'll understand that other 40% someday. Here's the thing that blows my mind. I'm into viruses, and it turns out about 8% of our genome is actually virus DNA. So we call these things endogenous retroviruses. I'm not going to go into all the details about what that means. Endogenous means in your genome. Retrovirus is a certain class of virus. And yeah, so big part of our genome seems to be composed of virus genomes. Very cool stuff. Worth a huge discussion, but I'm not going to go into that today. I think it's just very, very fascinating. So the viral component is approximately the Encyclopedia Britannica, by the way. So we also have our mitochondrial genome. It's not very big. It's 16,000 base pairs. So the plasmid that we extracted in the biology lab is about half the size of that. I think maybe about 7,000 base pairs or something like that. And this includes 38 genes. So you can see a bunch of these genes here are coding for actually transfer RNA. So I don't know if we have 20 of them for the 20 amino acids. I actually haven't counted. We have some of these things here are actually enzymes involved in either the electron transport chain or the citric acid cycle. You can see all of these here are actually complexes part of the electron transport chain. And that's about it. So it doesn't have everything that a mitochondria needs, only 38 genes. The citric acid cycle, most of those enzymes are actually imported. And the mitochondria does do other things besides the electron transport chain. But 38 of those genes are coded inside the mitochondria. So kind of cool. Chloroplasts, same thing. A bunch of genes are made in chloroplasts. Chloroplast genomes tend to be a little bit more variety there, and larger. You can see this one here that I found on the internet is 113 genes, and you can see it's coding for a whole bunch of things. We've got things like Transfer Rna. We've got photosystem 2 proteins, RNA polymerase, ATP synthase. So a bunch of things that are needed inside the chloroplast. So I think mitochondrial chloroplasts continue to be very fascinating organelles. So that's what a genome is. It's all of the DNA, right? And I made this little table for you that's kind of discussing a few things, and you can come back to it after we cover a little bit more lecture material. But this is kind of summarizing some of the things that I talked about, right? So, for example, eukaryotes are much bigger than prokaryotes, not just in size, but also in their genome size. Much of our DNA is non-coding, and often we have 2 copies. We're diploid, or like I said, in the case of some plants and some reptiles and insects, they can actually be more than diploid. There's tetraploid, there's hexaploid, all sorts of weird things out there. I think bananas have 5 copies of the genome, at least domestic bananas anyway. Our chromosomes are a little bit more complex. They have structural components that include telomeres and centromeres. And because they're so big, we also have histone proteins that help to spool the whole thing. Now, I know I told you before that prokaryotes do not have histone proteins. It wasn't entirely the truth. Again, here's another case in biology where we can just say usually. It turns out that some archaeal species actually have histone proteins, or something that looks like histone proteins. So interesting. We have organelles that have genomes, prokaryotes do not. And there are other things here that we haven't quite talked about in terms of regulation, these introns and these promoters and things like that. And we'll get into this in the next few units. A lot of those complex things are found in eukaryotes and not prokaryotes. So of course, promoters are found in all of them, but we haven't talked about promoters yet. That will be next week. So we talked about genomes. Now, what is a gene? Okay. So you're probably thinking a gene is something that gives you a trait, right? So maybe you have blue eyes or brown eyes. Maybe you have, some other trait that you've inherited from a parent, right? And that's true. That is one kind of definition of a gene. So you can see I have here, it's a discrete unit of inheritance. But another way to define a gene, and this is probably the best one here, is a specific nucleotide sequence. And often it's coding for a polypeptide. So you might code for a pigment in your skin or a pigment in your eyes, those kind of things. But usually it's a region of DNA coding for something. It doesn't necessarily have to be a protein. Remember that sometimes genes may code for RNA molecules like transfer RNAs. So like I said, time to expand your definition of a gene and start thinking about what it might actually code for. So here's kind of a typical eukaryotic gene. Notice it has exons and introns, and we'll get to that, what those are. But that's the coding part of the gene. The gene is going to have a bunch of regulatory stuff before, and it's going to have some regulatory stuff after. So notice we call this upstream and downstream. And I know this diagram looks really complex, and don't worry about it yet. We're going to get into these things. Like I said, this unit is just kind of getting you ready for the next couple units. So the gene is, that's made of DNA, and the DNA is going to get transcribed, and we're going to make RNA. So we're going to talk about making RNA, and that process is called transcription. The RNA itself can get processed, and sometimes it gets modified afterwards. And all of these things we're going to talk about next week, about what all of these things are. So just trying to show you the big picture here, and we'll tease apart the pieces one by one, starting on Monday. So we had talked about DNA, I think that was last Friday, where we were talking about Watson and Crick and how it was discovered that DNA was the genetic material. But it turns out that proteins are a more functional material, right? They code and they make things. that they can do a variety of functions, right? So we've got enzymes, we've got proteins that transport things across membranes, motor proteins, structural proteins, and so on. So I want to talk a little bit about kind of this concept of a gene. This is post-Mendel, where we're starting to try to understand the molecular mechanism of what exactly a gene is. So this goes back to some observations by this guy here, Archibald Garrod. And he was a doctor, and he was very interested in hereditary conditions. So diseases that were inherited within families. So this was one disease he was looking at in particular. It was a rare disease. I'm not exactly sure how to say that. Alkaptoruria. I'm not saying that right, I'm sure. But this is a condition, and people are lacking an enzyme in their urine metabolism. And so when they urinate, their urine turns black. So kind of a weird condition. And he realized that people who had this disease were lacking an enzyme. So he hypothesized that a gene is an enzyme. So this was his hypothesis. And actually, it's not too bad because a lot of genes do code for enzymes. And that's one way to explain our physical traits, right? So I want to bring this forward a few more years and get this, this hypothesis a little bit more solidified. So enter these two guys, Beadle and Tatum. And they were looking at mold, and they were hitting mold with X-rays. So the mold they're looking at is called Neurospora. It's just common like bread mold. It's called Neurospora because it looks a little bit like neurons. Very beautiful looking mold under a microscope. So they were hitting mold with X-rays. So just take a look at the left side of this for a moment. And you can see that you've got these normal wild type cells here. They hit it with the X-rays, and then they're getting mutants. So they're mutating mold. Okay, so at the time, there wasn't a lot understood around this mechanism, but we know now that this was damaging the DNA. But when you damage DNA, that can lead to damaged genes. And so what they were doing is we're looking at this here, this arginine synthesis pathway. So this arginine synthesis pathway was not quite worked out at the time, but they were working to this understanding. You start off with some precursor, you go to a second compound called ornithine, another compound is called citrulline, and the last compound there you can see is arginine. So it's a chemical pathway, and now we know that it takes 1, 2, and 3 enzymes. So basically they hit this mold with some X-rays, and they started looking at the mutants. And so they wanted to see what these mutants could grow on. And maybe it makes sense a little bit more in the next slide after this, but you can see that what they're trying to do is basically over here, they're feeding the mutants and finding out that they had different mutants that could live on different types of media, right? So here's kind of an explanation right here. So if you take a look at the wild type, right, and you think about that pathway, so minimal media is a very basic media that doesn't have a lot of food in it, right? It just has the bare minimum needed to survive. So the wild type can grow under the minimal media. It can grow if you feed it a little bit of ornithine, a little bit of citrulline, and a little bit of arginine. So the wild types are fully functional cells. They can grow with the bare minimum. They had one set of mutants that were called class 1 mutants. So the class 1 mutants could not grow on the minimal media. You had to feed them at least ornithine, citrulline, or arginine, but they did not grow under the minimal media. Turns out they had a second class of mutants that could not grow under the ornithine, right? They could not grow under the minimal media or the ornithine. And then they had these class 3 mutants that couldn't grow under any of these things, and you had to feed them the full amino acid arginine. So they started thinking about this, and they realized that they had actually solved what was going on in this chemical biological pathway, right? So I'd already showed you this picture here, and you can see under the wild type that we have a precursor. Precursor is converted to ornithine, and then citrulline, and then arginine. and that this took 3 enzymes. They hypothesized that the class 1 mutants were missing one of the enzymes in this pathway, and the class 2 were missing a different enzyme, and the class 3 were missing another enzyme. So they elucidated this pathway, and this really was their hypothesis, that each of these enzymes was a different gene. So they came up with a hypothesis, and their hypothesis that one gene is one enzyme. And that was it. And this was revolutionary to understand how it is that information in a genetic code can actually lead to a phenotype, right? So this is a phenotype they're observing in the yeast. Later, people kind of revised it to encoding for one polypeptide, because not all proteins are enzymes. Some proteins are structural, like they make your hair and keratin in your skin. They're not catalyzing any chemical reactions. But like I said, huge breakthrough in terms of understanding what a gene is. So gene 1, gene 2, and gene 3. Okay, so quick test yourself question. It says here, which of these following statements does not describe a gene? So let's take a look. A discrete unit of inheritance. That works for me. I inherited my blue eyes from my mom. B, a DNA sequence that encodes for transfer RNA. Yeah, that works. That's a DNA sequence encoding for something. C is a DNA sequence encoding for a ribosomal RNA. Yeah, I'll take that. A DNA sequence that codes for a protein. Yeah, that works. A DNA sequence that codes for a telomere. No, that one doesn't work. That DNA sequence is entirely structural. It's not coding any information in there. So that's not a gene. So telomere, structural only. It's just a repeat of nucleotides to make your chromosome a little extra longer. So that is the correct answer. So let's talk about these processes here, right? We want to go from DNA, we want to make it to protein, and we're going to use RNA as an intermediate for this. And so there's the 3 processes. I'm going to come back to this diagram again and again: replication, transcription, translation. So I want to talk about each of these a little bit. So in bacteria, by the way, there are no organelles. So all this is basically done in the cytoplasm. And sometimes all at the same time. And I'll show you some nice pictures, I think, next day of kind of all this happening simultaneously. I meant to put it on this particular slide, but I forgot. In eukaryotes, a little bit more complex. We've got that compartmentalization. The DNA, of course, is in the nucleus, and the DNA does not leave the nucleus unless it is, of course, mitosis. But during normal day-to-day operations, the DNA stays in the nucleus. So the RNA is made in the nucleus. The RNA is going to go out a nuclear pore, and it's going to eventually find a ribosome. So maybe I'll just put a little note that this happens at the ribosome, and that's going to make proteins, or cytoplasmic proteins anyway. Some of the RNA is going to find ribosomes, and those ribosomes are going to be attached to the rough ER, and those ones, those proteins are going to be targeted to specific locations. So some of them are going to get secreted, some of them might go to the Golgi, some of them might go back to the nuclear membrane, and so on. So endomembrane proteins, I'll call them, because we're talking about sending proteins to endomembrane organelles. So we will kind of come back to this here a little bit later in terms of how eukaryotes do this, at least some of the basics. So let's talk about transcription for a minute here. So transcription is basically scribing, right? If you think, if you were, let's say you were taking my class and you were, weren't able to write fast enough, or you had a disability, you might hire a scribe, and a scribe is just copying things down. Or if you think about the medieval scribes copying down, biblical manuscripts, they're just copying. And that's basically what transcription is. You're copying, but not quite perfect copying, because we're going from DNA to RNA, but it really is the same language. It's the language of nucleotides. So, if you take a look here, this T becomes a U, G, G, and then more T's become U's, and so on. So it's the same code, just U's instead of T's, and one strand instead of 2 strands. And that's transcription. So the enzyme that does transcription is called RNA polymerase, and it's a little bit more versatile than DNA polymerase. It doesn't need the helicase and all those kind of things. So that's something we're going to talk about on Monday. Step 2 is translation. So translation, we are actually changing languages, right? We are going from the language of nucleotides and we are going to the language of amino acids. So it really is a new language. So translation is a good word for this. And you probably know that we're going to use 3 nucleotides are called one codon, and one codon codes for one amino acid. And this all occurs at the ribosome. So that will probably be Wednesday's lecture next week. So a little bit of history for you. There's 20 amino acids. How can this be done? And so we'll go back 1950s. Remember, we had Watson and Crick. So right here, this is Crick. And this here is Watson. And you may remember these guys as being the people who figured out the structure of DNA. So the very next question was, how does DNA do what it does? And so a bunch of scientists formed this super exclusive club. It was called the RNA Tie Club. And I think this was actually the 1960s. And I'm not exactly sure the dates. And there are only 20 members. So some of you are probably thinking, 20 members, that's clever. Why 20 members? Well, there are 20 amino acids. So 20 members max, and there were 4 honorary members. I don't know what an honorary member is versus a normal member, but because there were 4 nucleotides. And these people would meet kind of regularly. I think it was like once or twice a year, get together and just discuss this problem because they were trying to figure it out. And they couldn't crack the problem. It turned out to be quite difficult. Here are some things though that they were thinking about, right? They figured, okay, well, there's 4 nucleotides and 20 amino acids. So we can't just, it's not a one-to-one code. It's not 1 nucleotide equals 1 amino acid. So what about 2? Okay, if you think about all the possible combinations of nucleotides, that only gives you 4 to the 2. So that's 4 times 4. That's 16 amino acids. That's not enough. So they figured probably it was 3 nucleotides coded for 1 amino acid. But how do you figure this out? Right? This is the question. How do you do this? So turns out that this guy figured it out, and he was not an RNA Tie Club member, Marshall Nurenberg. And his experiment, it turned out, was actually quite simple in some ways. So what he did is he synthesized, he was a chemist, and he synthesized just a long strand of U's, so a bunch of uracils, and that was it. And then what he did is he took a cell extract. So cell extract is where you take some yeast and you digest away their cell wall, and you release the contents of the cell. And so if you think about it, the contents of a cell, you've got ribosomes, you have nucleotides, and anything else that might be needed for this process of translation. So he added this synthetic nucleotide to the tube, and then he analyzed what he got. And so it turns out that U, and so on, coded for phenylalanine. So the abbreviation for phenylalanine is Phe. And he got a protein that was made of a bunch of phenylalanines. And he did the same thing for a bunch of A's and a bunch of C's and a bunch of G's. And he figured, at least started to figure it out. More people started getting involved at this stage. And of course, when you're looking at the other codons, it was a little bit more puzzle work, but eventually the genetic code was figured out. And so here is the genetic code, all 64 codons, and a codon is the triplets of nucleotides, right? So I want to point out a few things around this genetic code. So first thing to know is that AUG, you can see I have it here highlighted in green. That is the start codon. So I'm not going to ask you to memorize the genetic code. In fact, on the final exam, I will give you a copy of it. But you should probably know AUG, because that's the start codon and the most important codon here out of all 64 codons. That also codes for an amino acid called methionine, Methionine, the abbreviation for methionine is MET. What else can we say about the code? There's also stop codons. These ones are also kind of important. So these are the stop and start areas of a protein. They tell the ribosome start adding amino acids here and stop adding it here. And so when we talk about translation, we'll get into the details on that. So one of the things to know is that the code is, we call it degenerate. So what do I mean by degenerate? That means that multiple codons will code for one amino acid. So if you take a look at that one I have highlighted in the middle, that's proline. And it turns out there are 4 amino acids for proline, or sorry, 4 codons for proline. If you take a look at this one up here, this is phenylalanine. There are only 2 for phenylalanine. If we look at leucine, there are 6 for leucine. I'm not sure why leucine is so popular. It's actually a more common amino acid, so maybe that has something to do with it, and so on. So sometimes when we're looking at the code, the third letter, in fact, for proline, it doesn't really matter what that third nucleotide is. If you have a CC, whatever, they're all code for proline. So sometimes that's called the wobble position, that third position, you know, when you talk to genetics types people. So another thing about the code is it turns out it is mostly universal. So what is true for elephants is true for humans and E. coli and woolly mammoths. It turns out that all organisms on the planet use this genetic code. And so that's pretty cool. Certainly life would be a lot more complicated if every organism had its own coding system. That would be really weird. So let's talk a little bit about the ribosome and a little bit about translation. So we've talked about ribosomes already, and you may remember that there are different ribosomes in prokaryotes and different ribosomes in eukaryotes. And so ribosome actually has 2 subunits. Let me just skip ahead to the next page for a second. No, 2 subunits. There's a large subunit and a small subunit. And the ribosome basically grabs the messenger RNA. So this is the messenger RNA shown in red. And it's going to basically align the messenger RNA with transfer RNAs. So I know we haven't talked about transfer RNAs yet. Like I said, this is all going to be more detail next week. Just a good idea to kind of introduce some of it now. So the transfer RNAs, they actually have an amino acid, and they are going to bring the amino acid over to the ribosome. And they're kind of the translation technology. They're adapters that can convert from nucleotides to amino acids. So I'll show you what a transfer RNA looks like. This is a transfer RNA. So it's an RNA sequence. You can see it actually is kind of interesting because it has some single-stranded components, and there are actually some double-stranded components to this RNA. So this RNA has a couple of features to point out. One is it has an amino acid attachment site, and 2, it has something called an anticodon. And I think I have, let me just take a peek here. Yeah, there we go. So the anticodon is going to recognize the codon. So I have an example here. So here's an anticodon. It's 3' AAG. So the anticodon is going to recognize the codon that's complementary to that, right? So remember that A pairs with T or U. So we're going to have UU, and then G pairs with C. So the codon is UUC. So it matches perfectly. It matches by hydrogen bonding. And so the anticodon down here is going to correspond to a specific amino acid up here. And like I said, we'll get into this a bit more when we talk about translation. And so this amino acid, by the way, is phenylalanine. So let's try just a little puzzle here. You're going to see something like this on the final exam. This here is just a very basic one. We're going to start off with the DNA on the final exam. So just a warning, you're going to have a big question around this eventually. Right now, just baby steps. We're just learning some parts of this whole thing. So AUG, ACU, and so on. So what you're going to do on the question is basically break that down into the codons. So you can see what I've done is I've broken it down into the different triplets. And then you're going to use the genetic code. So AUG codes for methionine. ACU, you got to find that on your genetic code. So ACU is over here, codes for threonine, and so on. So my protein is going to be methionine, threonine, lysine, glycine. And then what about this last codon here, UAA? Okay, well that is the stop codon, and there's no amino acid named stop. You don't need to write stop. I'm just writing it here to show you where we started and stopped, but you don't need to write that. It's kind of understood. You've got a beginning and end. We don't need to write stop at the end. So here's a little bit more about what your question is going to look like on the final exam. It's going to be more complicated than this. This is maybe 1.5 marks out of 5. Like I said, we're going to add some more to this as we go on over the next week or two. So you can see we have a DNA sequence. And it says, give the RNA sequence and give the amino acid sequence. So the first thing to always do is look for this start codon. So the start codon is AUG. Now remember, this is the RNA, AUG. So the start codon on DNA is going to be ATG. So in this case, I made it super easy. You can see it's right at the beginning, right here, start codon. So notice there is another ATG here. I put that one in on purpose just to make a point. That is not ATG. I know you're thinking it sure says ATG. No, it's not. Remember, for nucleotides, we always read 5' to 3'. So that AUG or that ATG is actually not ATG, it's actually GTA if we read that correctly. So always find your ATG, your start codon. So once you find your start codon, we can start making the messenger RNA. So messenger RNA is going to look like the DNA, and you're probably wondering which strand of DNA is it going to look like? It's going to look like the one with the start codon. The only difference is it's going to replace all the T's with U's. So you can see I converted my ATG to AUG. CCG, no difference there because there are no T's. TAC becomes UAC, and TCT becomes UCU. ATC, AUC, CTA becomes CUA, and TAA becomes UAA. So usually at this point, what I do is underline them or somehow, break them up into those triplets, so those codons, and then it's just a matter of reading these off the genetic code. So AUG is, of course, methionine. So I apologize if I'm going a little fast. Normally I do this on the board, but I made this animated version to do it virtually. And usually people are writing this down and figuring out, but you can always watch the video later if you need to do some practice on this. And by the way, I did put a practice question on Moodle. The practice question is with the final exam study guide, and you'll notice there's some things in there that you probably won't be able to do yet, but at least you can take a look at it if you're looking to get some practice on this. So CCG, I can look that up on the genetic code. CCG, I think, is proline. So proline. UAC codes for tyrosine, and so on and so on. And so that's what my protein looks like. Remember the UAA, look that one up, is of course the stop codon. I haven't put stop because I don't need to put the word stop. It's obvious that's the end. So there is, like I said, part of what one of your final exam questions is going to look like. So we're going to add some more information to this and do it next week. And hopefully we'll get you some practice on that. So these processes, replication, transcription, translation, are called the central dogma of molecular biology because this is what all the cells do. And these are really important processes, which is why it's really important that you need to learn them for the final exam because they're very important concepts in biology. So you can notice my note here, one gene is one protein. This applies for all living things. So this has practical applications in biotechnology. So we can take genes, and I've talked about where we've taken a gene from human insulin and we put it in E. coli, and we make this product here called humulin. The one on the left is showing some laundry detergent, and it has enzymes for lipases and proteases made to put into the soap that can break down proteins and grease and things like that. So we'll talk more about biotechnology a little bit later. I just wanted to show you a couple of pictures from the textbook. Here's some other examples of a firefly gene in tobacco, jellyfish gene in pigs. You're probably thinking this is weird. Why are we doing this stuff to these poor animals? When you have colored things, it's easy to observe these genes and the phenotypes, right? So this is one reason why they do these, more of proof of concept rather than making a useful product. There are lots of weird products out there. This, by the way, is a Photoshop. I was looking for some genetic engineering information. I found this banana fish and thought I'd share that with you. But I will show you some real genetically engineered organisms. So I thought this was a video. It doesn't seem to be playing. This is a type of fish called a glowfish. And apparently you can finally get these in Canada now. So I was going to look into these and next time I set an aquarium, maybe get one. So these are zebrafish, and the zebrafish have fluorescent genes from jellyfish. In fact, since they've made these, this company has actually done a bunch of different colors and a bunch of different species of fish as well. So kind of cool. These are just novelties, but we'll talk about more practical applications of biotechnology in topic 19. So I kind of wanted to finish off talking a little bit and relating this to Mendelian genetics. So by that I mean Mendel, right? So you remember Gregor Mendel, and he was studying peas, and you may remember these Punnett squares and how you have recessive and dominant traits. So I kind of wanted to show you a couple of videos to show you how this works in terms of how a gene codes for protein, which equates to a phenotype. So this is a little video. I'm going to press play, and it doesn't have sound, but I'm going to talk you through it. So you can see we have two alleles. Those are genes. The genes code for RNA. Ribosomes take those genes and they make proteins out of them. And those proteins, in this case, are actually enzymes. And so they're going to show you what those enzymes do. Those enzymes are actually creating a pigment for a flower. And so here they go. They're making this reddish pigment. And so we have enough enzymes that we're going to get a red flower. And there's the red flower. So this is a homozygous dominant. So in this case, we have two functional copies of the gene, and we get a red flower. So let's take a look if we have a homozygous recessive. So what happens in this case? So same thing, we have two alleles, and because it's homozygous, we have the same alleles, and we make the RNA, the ribosomes make the proteins fold up. But in this case here, these proteins are actually non-functional. So you can see they're looking unhappy there, right? And so no pigment is going to get made. So you can see the enzymes they're trying, they can't do anything. They're non-functional enzymes. So these non-functional enzymes make no pigment, and then so we get no red, and so we get a white flower. So here we have two phenotypes, and all has something to do with the protein structure, right? So what happens when we get heterozygous, so one copy of each allele? Let's take a look at that in the video here. So there's the DNA, and the DNA is going to get transcribed, and we are going to make RNA. And the RNA is translated at the ribosomes to make proteins. One copy is a functional allele, and the other copy is non-functional. So the only question now is, what is going to happen? So we still have functional enzyme, and so we are still going to get some red pigment. So in some cases, you might not get enough red pigment and you might get a pink flower. In some cases, you have enough red pigment and you're going to get a red flower. So in this case, you have just enough to have a red flower, and that's what's popping up here. So we have a heterozygous, but with a dominant allele there. Okay, so last thing for today, I want to do a Kahoot. This is a bit of a shorter one, but just kind of going over some definitions here. And I kind of want to talk about some of these definitions as we go through the Kahoot. So let me share this and load it up. And it's loading now. Okay, just give you another minute for everyone to sign in. Okay, let's get going on this here. Okay, so there are some difficult ones in here, I'm just warning you. Question 1, which of the following best describes a gene? Good job. 4 people got the correct answer. So remember, a gene is a piece of DNA and it codes for something, it has information in it. So coding for a centromere is not information, it's just a structural component. And number 1 is RNA. So good job on that one. Number 2, which of the following best defines a genome? Okay, good job again. Okay, I think they're going to get harder soon, and we'll have a couple difficult ones here in there. Chloroplast genomes are. Okay, this one's an easy one. Circular, you just got to go back to that slide. Remember, they're circular like prokaryotes and circular like mitochondrial genomes. Question 4, replication is the process where, so read carefully. Giving you 30 seconds for this one rather than 20. Okay, so a little bit harder on that one. So remember, replication we talked about this week. And so we have DNA. So DNA is being copied. Okay, so if you take a look at these answers here, DNA is not being copied to RNA, so not the green one, and not the brown one either. So what's the difference between the blue and the red? The red one is saying single-stranded. Remember, replication, we're going from double-stranded to double-stranded DNA. So replication, make sure you know what that is and the details about that. Okay, this one also read carefully. Okay, so correct answer is the brown one. So told you these were hard, partly because we haven't talked about some of these processes in any detail yet. But what I wanted to do is throw this out front because I know when we get to the final exam, some people are still confused. So giving you some warning, you need to know what the difference is between replication and transcription and translation and the enzymes involved. So transcription is where we're making RNA. So it requires an RNA polymerase. So that's why the red one is incorrect, even though they're similar, right? We're making RNA, so RNA polymerase is the correct enzyme. Two more. Last hard one. Translation is. Okay, good job on that one. So people know a little bit more about translation. Translation is a ribosome. It takes messenger RNA and makes amino acids, attaches amino acids together. Question 7, the genetic code consists of how many codons? Should be an easy one. correct answer is 64. Oh no. Only one person got that one. Ouch. Take a look at the genetic code. We were just talking about that, right? You have 3 times 3 times 3 is, or sorry, 4 times 4 times 4 is 64 for the genetic code. All right, let's see how the scores come out. Bronze for Tristan and silver for Levi. And somebody got 6 out of 7, and I thought so. Well done, Nina. Okay, that's the end for today. So hopefully everyone has a wonderful weekend, and I'll catch up with you on Monday. We're going to talk about transcription. Take care, and we'll